Good evening, everyone. Good evening. Welcome to our first MAPS of the fall 2021 semester. We're really excited. We have a great lineup for you this semester, and we are hoping that most people can join us live. We will post a recording of the talk later on. But if you are here live, you'll get to ask questions of the speaker in the chat, and we will try to collect as many as we can and ask the speaker after the fact. So feel free to post questions in the chat at any time. So just a couple of announcements before we start. We just want to let you know that in addition to MAPS talks, we do have some science colloquium talks going on at MJC. Our first one is next Wednesday, Plant Pathology, Past, Present, and Future. So we have some talks for science colloquium. If you go to the MJC Instruction Science Math Engineering site at our MJC website, you can find the talks there. And our next MAPS talk is going to be next month in about battery, how AI can design better batteries, and that'll be Friday, October 22nd. So that'll be coming up in about a month. And then also I wanted to remind you that the Great Valley Museum is open. We're open currently Wednesdays through Saturdays, and there are planetarium shows every other Saturday. So we are starting to get more open here at the Great Valley Museum. So stop by if you get a chance. It is free for students, and check it out. It's really nice. Our speaker tonight is Gokul Ramadoss. He is a PhD student at UCSF. He has two advisors. He's co-advised by Martin Kampman and Bruce Conklin. And he has won some National Science Foundation Graduate Research Fellowship, and also recently was named a UCSF Discovery Fellow. And that is a PhD student chosen to serve as an ambassador for basic science education because of leadership potential, excellence in research, and communication skills. So we are really pleased to have him with us. And our talk tonight will focus on the development of safer and more precise CRISPR tools for therapeutic, genetic, and epigenetic editing. And he's especially interested in applying these techniques to treat neurodegenerative diseases that affect the brain and spinal cord. So welcome, Gokul Ramadoss, and we're glad to have you with us.
Thank you. Let me share my screen. Let's make sure. Does that look good? All right, cool. Thanks, everyone. So yeah, as Professor Linda said, I'm Gokul. I'm a graduate student studying the use of therapeutic gene editing to treat diseases that affect the brain and the spinal cord and basically anywhere that there are neurons. I'm recovering a little bit from a non-COVID illness, so I may be taking some sips of water throughout, but I'll try my best to power through. So yeah, I think a good summary of my research is that we're trying to use gene editing technology like CRISPR to treat genetic diseases. Excuse me. But for a lot of people, I guess the question is like, what does that even mean, right? What does it mean to edit a gene? What would gene editing look like as a treatment? So for this talk, I figured it would be most useful to talk more generally about what CRISPR is, how gene editing works, and how scientists like us are planning on using it in future medicines. And at the very end, I'll show you a little bit of recent data from our lab. But for the most part, my goal is for you to come away with this talk with three things. What is CRISPR? How does it work? And how can it be used to treat diseases? So let's start simple. Our bodies are made up of different kinds of cells that each have specific jobs. For example, there are different kinds of heart cells that make up your heart, and there are different kinds of brain cells like neurons that make up your brain. So if there's a heart cell whose job it is to beat, then it needs a lot of long fibrous proteins that can contract to help the cell beat. And if there's a neuron or a brain cell whose job is to communicate, let's say with your hand and make your fingers move, then the neuron needs proteins that allow it to send chemical and electrical signals all the way down to your hand in order to communicate. So whatever job that cell has, the proteins inside that cell are what allow the cell to do that job. And DNA is basically the instruction manual that tells the cell how to build all of the proteins that it needs. So for example, if your body was a house made up of all these different rooms, these different cells or organs, what allows those rooms or cells to actually function is the furniture inside them. For example, your bathroom wouldn't be a very good bathroom if it didn't have a toilet or a sink to use inside there. So the proteins are like the furniture that actually do the important jobs inside the cell and give it, like, define what the cell is and what it does. And the DNA is like the IKEA instruction manual that tells all the little workers inside your cell how to build each piece of furniture, each protein that it needs. So if you take all the different instruction manuals that IKEA has on how to build all the different kinds of furniture, and you staple them all together into one giant packet, that packet is called your genome. And each actual set of instructions, each instruction manual in that packet is called a gene. So here we are zoomed in on a part of the genome that has 4 genes from left to right: the genes for making a sink, the gene for making a toilet, the gene for making a TV, and the gene for making a couch. Something that a lot of people outside of science might not realize is that almost every cell in your body contains the same genome, which means that every cell in your body, no matter what it actually is supposed to be doing, is given the entire packet with every single gene. So every cell is given instructions on how to make every single kind of protein. You imagine if every single room in your house had every single type of furniture, right? There's like a TV shoved in your closet, there's a toilet in the middle of your kitchen. Your house would be so clogged up that nothing would work anymore. And we don't want that happening inside our cells. So the cell needs a way to know which genes it actually needs to read and which ones it can ignore. That way, each cell is only going to make the proteins that it actually needs. Our cells do this by turning certain genes on or off, depending on whether or not it needs to make the thing that that gene is coding instructions for. So there are some proteins that are required by every cell for general processes like breaking down nutrients or growing. So every cell probably has those genes turned on. But other proteins that do more specialized jobs are only required by certain cells. For example, your brain cell probably doesn't need to beat in the same way the heart cell does, so it doesn't need those same proteins. So in this example, in your bathroom cell, the genes for sink and toilet would be turned on, but the TV gene and the couch gene would be turned off. And that's okay. All that means is that the bathroom cells will build the sink proteins and the toilet proteins, but they won't build the TV proteins or the couch proteins. But in a living room cell, the opposite would be true. The sink gene would be off, the toilet gene would be off, but the TV gene would be on and the couch gene would be turned on. So in a living room cell, that cell would be making the TV protein and making the couch protein. So that brings us to genetic diseases. Genetic diseases are caused when there's a mutation in the genome, when there's a typo in one of the instruction manuals that your cell is reading. So if there's a typo in your instruction manual, excuse me, sorry, that means that your protein will be made incorrectly. And sometimes that's not a big deal. Like if it was just a mistake that makes your TV not able to turn on anymore, that wouldn't really cause any harm. You just won't be able to use your TV. But what if it's a mistake in the wiring of your TV that can make it catch on fire, right? That would be potentially a catastrophic mistake. So in that case, your whole living room, that whole cell could be damaged. Your bathroom, on the other hand, is probably fine because your bathroom cell probably has this gene, the TV gene, turned off because you don't need a TV in your bathroom. So since the bathroom cell has this TV gene turned off, it won't even read these instructions, so it's fine. But any cell that has this gene turned on, like your living room or your bedroom, for example, will read this gene with the typo. So it'll make the mutant form of this protein and will probably end up dying. In this example, it's catching on fire. And that damage could end up destroying the house or the entire organ or the entire human body, right? That's what we call it, a disease. And that's actually what happens in a lot of genetic diseases. The mutation in the DNA creates a mutant form of the protein that's now doing more harm than good. And that can end up damaging the entire cell, in this case, the entire neuron. So if this is a gene that's turned on in all of your neurons, then every neuron will read that gene with the typo, make that mutant protein, and eventually die. And that's what causes the brain to degenerate in these diseases, which is what we call a neurodegenerative diseases, because it's associated with the degeneration of neurons. Some examples of this that you might be familiar with are Huntington's disease, some forms of Parkinson's disease, some forms of Alzheimer's disease, and a few others that we'll talk about later. So how can we treat these genetic diseases? There's a lot of different approaches, right, at each of these different stages. At the body level or the organ level, people have come up with really cool therapies like physical therapy or even transcranial magnetic stimulation. These are ways to try to rewire the brain to compensate for the neurons that are dying in the disease. Excuse me. So these therapies are not likely to cure the disease, but they might help the patient cope with the disease better or cope with the symptoms of the disease better, slow down the progression. At the cell level, you could maybe say, we're in this disease, we're losing this particular kind of neuron. Can we regenerate that by making that kind of neuron in the lab and then transplanting them back into the brain? So the idea there is, can we replace the lost cells to restore the lost function? At the protein level is where a lot of drugs work. And the idea here is, can we just design drugs to interact with the dysfunctional protein, either to get rid of that protein or to change its shape to fix it or things like that? A lot of drugs are working at this level, and some of them are exciting. Some of them for some diseases just don't work. But what we're talking about today is how to treat genetic diseases at the genetic level. We want to address the root cause of the disease by correcting the DNA mutation itself, or at least by turning off the mutant gene to stop producing the mutant protein. So it's exciting that with CRISPR tools, right, we have the ability to address these diseases at the root cause. And hopefully, by correcting the mutation, we can halt the progression of the disease, or maybe even prevent or reverse it. That remains to be seen. So the three topics we'll cover in today's talk are: one, what is this word CRISPR I keep hearing about? Two, how do we use CRISPR to edit genes? And three, how can we use CRISPR in gene editing to cure genetic diseases? So to understand what CRISPR is and where it came from, we have to look at this bloody battle that's been going on for millions of years, the battle between bacteria and viruses. There are viruses that have evolved to infect bacteria, and those viruses are called bacteriophages, meaning bacteria eaters, basically. So bacteria are afraid of bacteriophages, and they're constantly rapidly evolving new tricks to try destroying those viruses. Meanwhile, the bacteriophages, the viruses, are always evolving new tricks to evade those defenses and still get into the bacteria. So in this constant battle and constant arms race, there's a lot of cool evolution that comes out of it, as both sides are trying to evolve new weapons to use against each other. CRISPR is one of those weapons. And to be specific, it's a weapon that bacteria evolved to protect themselves against these viruses. So how does that work? Let's say this bacteria was infected by this bacteriophage or virus, and that virus injects its DNA into the bacteria, because that's how viruses try to reproduce. The bacteria then uses some special proteins to take a mugshot of what that virus looks like by capturing snippets of that viral DNA or RNA. And the bacteria stores those mugshots in its own DNA. That way, by recording this information permanently in its own DNA, that way, even after its own lifetime, when this bacterial cell is reproduced and passed on its DNA for generations and generations, its descendants will still have this record in the DNA of what kinds of invaders have tried to infect it before in the past. So this really creative way of storing those snippets of genomes from previous viral invaders is CRISPR. CRISPR stands for clustered, regularly interspaced short palindromic repeats. You don't need to know that. It's basically a fancy way to describe this uniquely patterned DNA region in green that people found when they were studying bacteria and trying to understand what it did. And this unique DNA region was interspaced by these mugshots of these previous viral invaders, things that had previously tried to infect the bacteria. So the bacteria is collecting those mugshots of viral invaders, recording those mugshots in its DNA, and passing them down to future generations, but it's doing all of this for a reason. Each of these bacterial cells then puts that mugshot onto a wanted poster and makes tons of copies of that wanted poster throughout the cell. These wanted posters are picked up by proteins that act like bounty hunters, patrolling the cell and looking for the criminal, right, this virus. And if that same virus ever tries to infect the same cell again, or its descendants, then these bounty hunter proteins immediately recognize it based on the wanted poster because the sequence matches, and they destroy the virus by making a cut. So this is one way that bacteria have evolved to protect themselves against viruses. Since they don't have antibodies like we do to protect against viruses, one trick that they've evolved is this CRISPR system. One of these bounty hunter proteins that some bacteria have used is called Cas9, which stands for CRISPR-associated protein 9. And that's the one most of you have probably heard of, and it's the one we'll talk about today. So there are many different labs involved in the discovery of what CRISPR is and how bacteria use proteins like Cas9 to cut DNA. But Jennifer Doudna and her lab and her collaborators made the breakthrough of taking the system from bacteria that bacteria were using to protect themselves against just viruses and engineering it into a tool that we can use. And this discovery won a Nobel Prize last year. The bacteria would use Cas9 to cut virus DNA that matched its wanted posters. But for us, thanks to this really cool engineering that they've done, we can swap out the wanted poster for whatever sequence we wanted to cut. And that lets us use CRISPR-Cas9 as a tool to essentially make a cut in whatever sequence of DNA you want, including making a cut in our own human genome, not just viruses. So I guess before I move on to how we use CRISPR to edit genes, we can take a couple of questions just in case anyone has some clarifying questions on that first part.
Okay, so we had a really good question from Jeff. Jeff said, what are the pros and cons of DNA editing versus messenger RNA editing?
That's a great question. Can we come back to that at the end?
Okay.
Okay, thanks. I think the pros and cons will be more of, will involve the therapeutic aspects that we'll talk at the end. That's a great question.
That's the only one I have for the moment.
Okay, cool. Great question. So, like I said, Cas9's job is, in our case, is to make a cut. And it sort of ends there. In this diagram here, the yellow protein is Cas9, which is basically a protein that we use to cut DNA. The orange strand there of RNA is called a guide RNA. You might see it called sgRNA or single guide RNA. You just call it a guide RNA. And that guide RNA is the wanted poster that tells the Cas9 where to cut. Thanks to people like the Doudna lab, we get to decide where the cut is made by programming it in, because now we're the ones designing that wanted poster. So we just designed that guide RNA in orange, which is a sequence of 20 or so letters that matches the sequence of the DNA that we want to cut. If we want to cut the sequence like AAAAA, then we just design a guide RNA that matches that. We can order it and from like many different companies, and we can just get that guide RNA within like a week. It'll arrive. So we can design a guide RNA targeting whatever sequence of DNA we were interested in, order it, receive it pretty soon. Then we just have to mix the guide RNA with the Cas9. And now we have the Cas9, which can cut DNA, bound to a guide RNA, right, this wanted poster that's telling it where we want it to cut. So if the guide RNA sequence matches or finds its matching DNA sequence inside the cell, then the Cas9 makes a cut there. So how this actually works is when there is a full match, the guide RNA sequence matches the sequence at this part of the DNA, it causes a shift in the form of the Cas9 that allows it to move from an inactive conformation into an active shape that then opens up these two sites for the Cas9 to make a cut. So it basically cuts both strands of the DNA, and that's about it. The Cas9 makes the cut. Once the Cas9 makes that cut, what happens next really depends on the cell. Right, we've talked about so far how to use Cas9 to make a cut, but that's only part of the story because now we need to actually decide how to edit the genes, right? Cutting is only half the story. So for this, we rely on the cells. Cells don't like to have breakages in their DNA, so they have lots of different DNA repair proteins that immediately get to work once this cut is made. One option is that the cell can just try to jam the ends back together again, but every time it does that correctly, if the target DNA sequence remains unchanged, then the guide RNA sequence on the wanted poster still matches perfectly with the DNA because you didn't change the sequence. So it cuts again and again and again until eventually the cell happens to make a mistake when it's jamming the ends back together. It either ends up missing a few letters or accidentally adding in a few extra letters, and the gene ends up being inactivated. Only then can the Cas9 no longer cut there because now the sequence has been changed, so the guide RNA doesn't match it anymore. So it's done cutting. This gene being inactivated could actually be a good thing if we're trying to inactivate one of those mutated genes that's producing a toxic protein. So we can use this method to shut off the mutated gene. I should also mention that even if Cas9 never had any applications in medicine in like curing a disease, the fact that you can use this to really efficiently turn off genes is in itself a huge biological breakthrough. And honestly, that's probably why this was like a Nobel Prize winning breakthrough, because scientists, like a lot of people, like to learn how things work by breaking them, right? If you want to know what gene B does in a cell, the best way to do it is to turn off gene B in a bunch of cells and see what changes, right? So people have used Cas9 not only to turn off one gene at a time, but to turn off lots of different genes at the same time and learn what a lot of different genes do. So the fact that you have this really efficient way to turn off a gene is in itself very powerful. What people like me work on is using that ability to turn off a gene that's causing a problem in a genetic disease as well. But I just wanted to throw that out there, that even in just studying biology, it's already a very powerful breakthrough. So there's another way that the cell can repair this cut. If it can find a template of what the sequence should look like, it can try rewriting the letters around the cut and correcting the mutation. So people can try to actually provide the cell with the template of the correct unmutated sequence. And sometimes the cell will like rewrite the mutated sequence that you've cut into the correct healthy sequence. But rewriting genes tends to happen a lot less efficiently compared to inactivation, just an FYI. So inactivation is often quite efficient at turning off the gene that you want to turn off, which is why in the strategies that I'll talk about today, we're going to focus on the inactivation strategies. There's an analogy that I really like from Feng Zhang, who's another scientist who was also instrumental in engineering Cas9 for human use. And he described Cas9 as like a cursor that you would use in Microsoft Word. So I wanted to combine these two analogies, right? So let's say we want to fix that mutation in our IKEA manual that's making our TV catch on fire. So we open that document up in Microsoft Word, and we see the instructions in this gene are supposed to say, plug wire into blue port. But instead, the mutation in this gene accidentally says, plug fire into blue port. So we have to fix this. So we would use Command F or Control F to search through the document for the word fire, right? F-I-R-E. That's our guide RNA doing that searching, specifying what sequence we should look for so the Cas9 can go make a cut there. So once we find that location of the document, F-I-R-E, fire right here, we click and we place our cursor right there, right by the F. That's all the Cas9 really does. It just makes a cut. From there, if we want to try inactivating the gene, then we want the cell to make a mistake when trying to repair this cut. So in this case, it accidentally deletes that letter F. So when the cell tries reading the instructions to make a TV, it goes word by word, but all the letters have been shifted over because one letter was missing. So the words actually don't make any sense anymore. And the biological term for this is literally called nonsense. So the cell realizes that this gene has become nonsense, so it just stops reading it. It's no longer making those flammable TVs. The other strategy would be to try making the cell replace the mutated word, right, the word fire, with the word wire. So in this case, you would provide this template of the word wire and hope that the cell incorporates that word wire in the rewrite and hopefully corrects the mutation from fire to wire. And in theory, this would be a more ideal outcome because you're not just turning off one of the genes, you're actually fixing the gene entirely. But as I said before, this tends to be, at least with current strategies, it tends to be a little bit less efficient than inactivation. So for the rest of this talk, I'll focus on the inactivation strategy. But like I said, there are a lot of really exciting CRISPR-based technologies being developed to make rewriting genes more efficient as well. We just won't talk about this today. Today is for the basics. So I'll pause there. We've talked about how we use CRISPR to edit genes by specifically inactivating them after we've made a cut. Are there any questions on what I've covered so far before we move into using it to cure genetic diseases?
Yeah, I've actually got a whole slew of them here. Let me go up to the top. We're holding a question on pros and cons. So one person asked, when you say reversal, you mean in adults? And so I assume that they meant using CRISPR to reverse a condition, because there are a couple of questions dealt with, or is this something we can use with infants or children? So they're concerned about the adult versus child application.
Mm-hmm, mm-hmm. Yeah, that's a good question. So I should say, I said hope. I meant to say like hopefully reversal. We don't know if it would reverse anything. We hope that this would at least halt the progression of the disease, stop it from getting worse. So if that is the case, and if it doesn't reverse the disease, then yeah, it would be more efficient in children or younger adults compared to older adults, in which like too much damage has already been done. Whether halting the disease at a certain stage is possible or reversing it at a certain stage is possible probably depends disease to disease. That's a very good question.
Did you want another?
Sure.
So we took care of that. So there was a couple of questions on the oversight or regulation of CRISPR. Specifically, how do you know what you purchase online is reliable or safe? And I wasn't aware that it was available online for treatment.
I see. Oh, that's a good question. So I'll answer that in a way that doesn't touch on the therapy yet. I guess, how do you know that what you receive is good quality? I mean, I guess it's the same way when you're using it for a biological tool. It's the same way that you would trust any other reagent that you would order in the lab. We order lots of different proteins and lots of different sequences of DNA and RNA, and you would use the same tools in that case. Like if your experiment just didn't work, then you could look at maybe like if you would try buying the Cas9 from another company and it works better, right? That could work. There are definitely techniques that people who are looking into this could use. Like they could look at, are the amino acids that you expect to be in that protein actually there? Or does the sequence of the guide RNA actually match what you were expecting? But for our case, we just have suppliers that we trust and we would just use those. But good enough for biology is definitely not the same as good enough for therapy. Like you probably wouldn't just order it online from one of these companies and use it for therapy, and shouldn't do that. Yeah, what I'm talking about is when I say like you can order this and get it in like a week, I meant to do an experiment in the lab, not on a human, just in cells.
More questions or?
Yeah, I think we can take a couple more.
Okay, so a couple of concerns with the ethical aspects of genome editing. How do you navigate and address the ethical aspects of genome editing?
That one I think we'll talk at the end.
Okay.
Once we talk about like what we can do with it therapeutically.
So I'll skip down to the next one. When you edit the sequence, is it with an actual cell from a patient, or is it just the strand on its own?
Good question. Both are possible. So some of the early experiments showing that you could do this started off with just editing a strand of DNA in a tube. But since then, people have shown that this works in cells as well. And a lot of the experiments that I'll show you and the data that I'll show you is all done editing in actual cells. Not necessarily in the data I'm showing is not like in people, but it's in cells that we've gotten from people, or cells that you can just grow in a dish in the lab. The challenge there is that obviously it is harder to do editing in a cell compared to editing just in a strand of DNA in a tube, for a couple of reasons. One is that there's a lot of other proteins moving around and interacting with the DNA and interacting with the Cas9 that you have to navigate, but also getting into a cell tends to be a challenge as well. And we'll talk a little bit about that in the next bullet, but we call this the delivery challenge. Like if you're just mixing Cas9 and guide RNA with the DNA in the tube, they just find each other, they cut. But in a cell, you have to actually get into a cell, that's designed to keep a lot of things out and then not get degraded and things like that. So getting the Cas9 into a cell is not necessarily trivial, but it is something we do regularly.
Another question or moving on?
Yeah, let's take one more question on this.
So what's the chance that it actually mutates the genes even more? In other words, making additional mistakes or taking out something that it shouldn't?
Yeah, another great question. So that is definitely always a possibility. So it's very important when you're proposing something like this as a therapy to develop really good assays to look at what you've done and make sure that you're doing what you're trying to do, right? So once we've made an edit, we'll often sequence the DNA of our cells to make sure that we know what the editing outcome was, whether we, first of all, made the edit that we wanted to, but second of all, did we accidentally make any other changes that we didn't want to? These are very important things. And honestly, among some of the biggest challenges facing the field as we go forward to a therapy would be the safety and precision of these edits, improving those, as well as improving the delivery, like I said.
Yeah, because our next question, or one of the next questions was, what are the big risks of this?
Yeah. So that's one of them. I think we can talk at the end about the pros and cons. But yeah, these are all great questions. So like I said, safety, efficacy, precision, these are things that are at the top of everyone's minds as we're going. And people continue to develop better and better assays for detecting, all of these potential unintended edits, making sure that your edit is actually doing what you want it to do. These are all very important things that even when we're just doing it in cells, we often have to verify these things to make sure that the biology that we're studying is actually what we meant to study and not just an effect of just making a random unintended edit.
Okay.
Cool. So let's talk about how we can use this gene editing tool now to do editing in a patient to try to cure a genetic disease. In some tissues, like the blood, for example, it's possible to take the cells out of the patient, edit them, edit their DNA, and then put the cells back into the patient. And in those cases, it honestly might be a little bit easier to, to treat those diseases because you can take out a lot of cells, edit them, and then put them back. So a lot more strategies, editing strategies are available in those kinds of therapies. However, in tissues like the brain, these cells can't be taken out and put back. You can't, like, it would be very hard to take the neurons out of a person's brain, edit them, and then put them back and expect them to be okay, right? So in those cases, we have to deliver the Cas9 and the guide RNA directly into the patient. We can't edit the cells outside the body and then put them back. So this is a challenge called in vivo gene editing, doing the gene editing in the person's body itself. So in that case, it's likely that rewriting the genes likely wouldn't happen in enough cells. So this is why in the therapies that I'm talking about in the rest of this talk, we're again going for the inactivation strategies. Because let's say this rewriting therapy was very inefficient. You were able to rewrite like 10% of the cells, or actually that's pretty efficient. If you're able to rewrite like 1% of the cells, if this was a strategy where you could take like the cells out of the body, edit them, and then select for the 1% that were edited, make a lot more of those and put them back, then that 1% efficiency is pretty good. But in the case of neurons, where the only neurons that you're editing are the ones that are already in your body, if you're only able to, correctly edit 1% of them, that's probably not going to be enough to have a therapeutic effect. So for this talk, I'm focusing on the more efficient strategy, which is an inactivation strategy rather than the rewriting ones. Both are very promising though in the future. So in this case, the therapy is, if there's a mutant gene that's producing a toxic or harmful protein, like this TV that's catching on fire, let's inactivate that gene. Let's turn it off, like we did before, to stop it from producing that toxic protein. And what I'm going to focus on is motor neurons. This is one of the things we focus on in our lab, because this is a kind of neuron where there's a proven way of delivering things to them, which, like I said, is one of the biggest challenges in a lot of different cell types, is we don't have like particles or methods of getting stuff into them safely. Motor neurons are one of the first tissues that things like DNA and stuff have been safely delivered to in vivo. So we like that the delivery problem has already been partly solved in this case. And there's a lot of clear genetic markers. For example, if you were considering a disease like Alzheimer's, if you're trying to tell, whether someone or whether a mutation or a correction in a mutation is correlated with a benefit or not in their disease progression, it's a lot harder to measure some of those effects, like a learning deficit or a memory deficit, than it is to measure, like, can you walk, or how long can you walk, and things like that. So motor neuron diseases tend to have a lot clearer genetic ties, just because people have been able to do these very concrete assays. And because the genetics are clear in the motor neurons, it makes it a very good target for our genetic therapies, where we have a lot of data on, like, people with this mutation are okay, but people with this mutation are not okay. If we make this edit, it looks like these patients' genomes who are able to walk perfectly fine. So we know that this edit would be safe. Whereas with a lot of other diseases, we'd sort of just be guessing at whether, if we inactivate one gene and leave the other one, we'd be guessing whether or not they'd be okay. We don't really have that data. So for that reason, in our lab, we focus on motor neuron diseases, at least for now. But there's still a lot of motor neuron diseases that are genetic. So how do we narrow that down further? One way to filter this down is looking at only monogenic diseases. Monogenic meaning this is a disease that's caused by mutations in only one particular gene. So one of the diseases I mentioned, Alzheimer's, there's a lot of different forms of Alzheimer's, but some of those forms are caused by, like, either contributions from multiple different genes, or some of them are not even caused by genetic factors at all. So those would probably not be the best targets for these initial gene editing therapies, because you'd have to either edit a bunch of genes or like some really complicated strategy. Whereas for now, if this is a new kind of therapy, so we'd like to focus on diseases that can be cured by just targeting one mutation in one gene. Another filter is we would like these to be dominant diseases. And what dominant means as opposed to recessive is basically most people have 2 copies of every gene, one that you inherit from each parent. So if it's a recessive disease, that means it's a disease that's caused only when both copies of the gene are mutated. But if it's a dominant disease, that means it's a disease that's caused by having even one mutant copy, even if the other copy is perfectly fine. So our approach with inactivation is pretty straightforward for dominant diseases. We just basically have to inactivate the mutant copy and leave the healthy copy alone. That brings up the next point, though, of haplosufficiency. Basically, this means that our cells have to be okay with only having one copy of that gene left. Haplosufficient, meaning half of the normal amount is okay because you're inactivating one copy. For some genes, this is not the case. For some genes, your cells actually need both copies to be active. Those genes are called haploinsufficient because having half the normal amount is not enough. So if we're going to inactivate one copy of your gene, we have to have evidence that that gene is haplosufficient, meaning we have to have evidence that people who have just one active copy of that gene are okay. So this is where that clear genetics of motor neuron diseases compared to other diseases really comes into play. We're a lot more confident that in some of these motor neuron diseases, people are walking around perfectly fine who only have one active copy. And we've seen this from human genetics. Another thing is this has to be worth the risk. And this touches on some of the ethical questions, especially for a new therapy that's making permanent changes in the DNA and things like that. There better be no other effective treatment options that are already available for this disease. Because if not, why would you ever justify doing such an experimental therapy, right? If most of the diseases that we're going after with these gene editing therapies are diseases that have no other effective or curative treatment options available. And the last thing is measurable outcomes. We want to be able to tell whether or not there's improvement, or else like in a clinical trial, it would be very hard to tell whether or not it worked, right? And this, again, is an example of the motor neurons versus other neurons. It's a lot easier to measure someone is more able to walk rather than someone's memory deficit is slightly improving or slightly worsening, and things like that. So we did this filter, and in the lab, we've selected a lot of motor neuron diseases that fit all of these categories. The first one is Charcot-Marie tooth disease, the one that I'll talk about today. Charcot-Marie tooth disease, or CMT. So how can we approach therapeutic gene editing for Charcot-Marie tooth disease? First of all, CMT, fun fact, is the most common hereditary neuropathy. Hereditary meaning it's a genetic disease that you can inherit from your parents, and neuropathy meaning it's a disease that affects your neurons. So it's actually the most common genetic disease that affects your neurons that you can inherit. There are many different forms of CMT. However, each of these different forms tends to be caused by mutations in only one gene. For example, there's CMT type 2A, which is caused by mutations in the gene MFN2. There's CMT2E, which is the one we'll talk about today the most, which are caused by mutations in the gene NEFL. And there's CMT type 2F, which is caused by mutations in the gene HSPB1. So how this disease works is basically it causes progressive degeneration of the motor neurons, starting from the longest motor neurons, and then increasingly as the disease progresses, more and more of the shorter motor neurons will be affected as well. So on this diagram on the right, the ones in red are basically where the motor neurons are. These are the lower motor neurons, so that means they live in the spinal cord, but they project out to the extremities. So the first motor neurons to be affected by this disease would be the ones that extend all the way from your spine down into your toes and into your fingers and things like that. But as the disease progresses, this paralysis spreads from just a foot deficit, like it starts with it being more difficult to like raise your foot at the ankle, for example, but it ends with increasing paralysis, people needing either walking braces and then scooters to get around and things like that. Many forms of CMT, like CMT2E, are potentially treatable with therapeutic gene editing with an inactivation strategy for two reasons. One is that some of these forms are autosomal dominant, which, as we talked about before, means that the disease is caused by the presence of one mutant copy, even if one healthy copy is still there. And these genes, we have really good genetic evidence, again, because of motor neuron disease, that these genes are haplosufficient, meaning there are plenty of people who lack one copy entirely, and they're totally fine. So in this case, the strategy is pretty obvious. We're going to try to inactivate the mutant copy and leave the healthy copy alone, because we know there are a lot of people out there who only have one healthy copy, one missing copy, and they're fine. So again, for most genes, you have two copies or two alleles, one that you inherit from each parent. An analogy for that I like to use is if you're trying to build a wall, but you're given two piles of bricks. For most people, this is not a problem. You just use both piles of bricks and you have a good wall. But for some people, from one parent, they inherit a healthy pile of bricks. And from the other parent, they inherit a diseased pile of bricks, right? So if this disease is dominant negative, that means that even a presence of a few of these bricks in the wall, these disease bricks, is enough to destroy the whole wall. So our strategy here is to get rid of that second pile of bricks entirely and force your cells to only use the first pile to build your wall. That way you don't end up poisoning the wall. So as long as losing half of your bricks still leaves you with enough bricks to make the wall still, right? So that's the haplosufficiency part again. If this was not a haplosufficient gene, then you actually need enough bricks from both piles to be able to build your wall. But in the diseases that we're focusing on, this is not the case. One pile is more than enough bricks that you need to build this wall. So this is why this bottom strategy is what we're going after. Just inactivate that mutant allele, and hopefully you're able to build that wall. So what does this look like in CMT type 2E, right? This is our example from before. This is caused by mutations in the gene NEFL. Obviously, people who have 2 healthy copies of this gene are fine. They have healthy motor neurons. But we know from human genetics data that people who have 1 healthy copy and 1 dominant negative mutation, because it's dominant, they have a very severe disease and they have this motor neuron degeneration. Luckily, we also know from human genetics data that, like I said before, people who have 1 healthy copy and 1 copy that's turned off and inactivated by some other mutation, they don't show any signs of disease. So this is very encouraging for an activation strategy because we have strong evidence that the genotype that we're going after, meaning the inactivation of 1 mutant copy but leaving the healthy copy alone, is correlated in patients with being perfectly fine in terms of your motor neurons and being able to walk and things like that. So how can we develop or test this gene editing therapy? How can we test whether it's effective, whether it's precise, whether it's safe? How can we try editing human neurons and seeing if it works, but without experimenting on a human? This is where stem cells come in. We're not going to experiment on humans, but we can use stem cells to create neurons in the lab and experiment on those. So what are stem cells? Very early on, just a little bit after fertilization, your entire body is just a few cells. And those few cells will eventually have to turn into every single kind of cell in the body. So a lot of those cells are called pluripotent stem cells, meaning they have the potential to turn into a lot of different cells in the body, almost all of them. So the stem cells are like little babies that haven't yet decided what they want to be when they grow up. And as the embryo develops, those stem cells will all receive different signals that guide them down the right path, like going to college, picking a major, taking all the right courses to become a doctor or a lawyer or an architect, right? So similarly, these stem cells will eventually differentiate or turn into all the different cells in your fully developed body, from the brain to the heart to the gut. So stem cells are a very powerful tool for us to work with in the lab, because if we know what signals to give them, we can turn those stem cells into whatever kind of cell we want. For us, that means neurons or motor neurons. So Shinya Yamanaka is the second Nobel laureate pictured in this talk. He discovered a way to make stem cells without requiring any embryos or anything like that. He discovered a way to take differentiated cells that have already turned into something like a skin cell or a blood cell, and then transform them back into pluripotent stem cells that can become whatever you want again. These are called induced pluripotent stem cells, or iPSCs. And you can make iPSCs from anyone, including from a patient who has the disease that you're trying to treat. So because these cells are coming from the patient, you can just take a skin sample from the patient or a blood sample. They just donate blood, and you can use those cells to make stem cells. Because these skin cells or blood cells or whatever are coming from the patient, they have the exact same DNA as the person that they're coming from. So they also have the disease that the patient has. So if we give these stem cells the right signals, we can actually model in a dish that patient's own neurons, what they look like and how they behave. And the mutation that the patient has is in these neurons as well. So we can actually compare neurons that come from patient iPSCs against neurons that come from healthy donor iPSCs and see how the patient neurons differ from the healthy neurons. And more importantly for us, we can then try out our different gene editing therapies on the patient's own neurons in a dish and see if we can reverse the effects of the disease, or at least halt the effects of the disease. So here's a time-lapse video taken by an RA who used to work in the lab, just so you can see that these motor neurons, right, they extend these really long axons as they grow. In your body, these would be even longer since they have to reach from your spinal cord all the way down to your fingers and to your toes. But even in a little dish in the lab, they're still pretty long. This is just to show you that, we at our lab and others have developed really good ways of turning stem cells into the kind of neurons or the kinds of cells that you want to test on in the lab. So what can we do with these stem cells and these stem cell derived neurons? This is getting into some of the recent data that our lab just published. since we can make neurons out of these iPSCs from patients or from healthy donors, we can then study how patient neurons differ from healthy neurons. So in this form of CMT, again, it's called CMT2E. The gene that's affected is called NEFL, and it produces the NFL protein, which stands for neurofilament light chain. So in the last video, we saw these really long projections coming out of the neurons called axons. These are the things that allow them to make connections, like reach all the way out of the spinal cord into the right muscle and make a connection there. So when it fires, it tells that muscle to contract. So it needs these really long axons. And because it has these really long projections, the neurons need to be able to transport things up and down the length of that axon from the main cell body. So neurofilaments are one of the main ways that, or one of the many ways, I guess, that neurons use to transport things along the length of their axons, almost like highways that are allowing the transport of important molecules up and down the neuron. So it's well documented that in the spinal cords of patients who have CMT2E, this neurofilament light chain protein, NFL, isn't being distributed properly along the length of the axon. And instead, it seems to be getting stuck and aggregating up here in the main cell body, if you can see. So in normal patients, it's nicely evenly distributed along the axons, but in normal people, in people with the normal sequence of this gene. But people who have a little typo in this gene, that minor typo has a huge effect where all of this protein that's supposed to be distributed along the length of the axon tends to aggregate right next to the main cell body instead. So it's clearly not able to do its job here. And in our iPSC-derived neurons that we make in the lab, we can see that same phenotype. That's actually what's pictured here. That same phenotype or that same symptom in the patient-derived neurons compared to the wild-type neurons, which means neurons without this mutation. This is quantified on the graph on the right. You can see that the amount of NFL protein that's stuck around the cell body instead of being in the axons is close to double what it should be in a healthy individual. It's pretty cool that we can see this disease symptom in the cells that we're modeling in the lab. But in addition, that means that if we can test our gene editing therapy, we can see if when we inactivate the mutant allele, whether that restores this symptom back to normal. And we see that that is the case. So on the right two images here are two different strategies for inactivating the mutant allele. For editing the mutant allele. One is the inactivation strategy that we would probably be testing in future experiments. And the right one is the little more, less efficient one, but where we actually try to rewrite or correct the mutant allele, rewriting the mutation to what the correct sequence should be. And in both cases, that effect goes away. That NFL protein is no longer sticking around the cell body and is more able to be distributed as it should be. And the graph at the right is just quantifying that, where in dark blue you see the wild type, the healthy neurons, what they look like. In red is the patient neurons, what they look like. And then both of these therapeutic strategies, both the inactivation and the rewriting, restored the symptom back to normal levels. That pretty much matched what the wild type levels looked like in the healthy individuals. This is very exciting, but what are the next steps for this, right? Because it's very different to deliver Cas9 to stem cells in the lab, turn those stem cells into neurons, and say, oh, these neurons look better than the ones where we didn't correct the mutation. It's a very different question from looking into the clinic and saying, what if I delivered this Cas9 directly into the patient's own neurons that have already developed some amount of the disease? Can I make the edit there? And will that then restore the symptoms in that patient, right? This is a completely different question. And this is sort of where the main focus of my PhD is. I'm trying to engineer safer, more precise ways to deliver Cas9 into neurons, again, maximizing effectiveness, but also safety and precision. And if we do end up engineering particles that work really well in neurons, maybe we'll come back here one day and talk about those. But that's one major thing, is how can we deliver Cas9 into neurons, not just into iPSCs or stem cells? How can we optimize the safety or precision of those edits, delivering the Cas9 in a way that we're not delivering like more than we need to, so that we're not increasing the chance of the Cas9 being up to no good and making all these unintended side effect edits after it's done the job that we wanted it to, as well as testing whether, if we can make that therapeutic edit in a patient directly or in the cells, in the neurons directly, instead of editing the stem cells and then turning them into neurons, we can then test that really important question that someone asked, which is, at a stage when the neurons have already developed and have already shown some symptoms of the disease, what happens if we edit then? Do we just stop the disease from getting worse? Is that good enough to save the neurons from dying? or could we even see some reversal of that disease progression? Maybe the neurons can actually get better over time as well. We'd only be able to answer these questions once we're able to really efficiently deliver Cas9 into our patient motor neurons, and that's basically the next steps that we're working on for the next couple of years. So I'm still in the early days of this delivery front, but I hope that this presentation has given you all a better understanding of what Cas9 is, how it works, and how we can use it to try treating genetic diseases, so that the goal ultimately will be to take diseased neurons, whether that's in a patient or in an animal model or in a dish, use this inactivation strategy or some other editing strategy to edit the genes and hopefully see that either reversing the disease phenotype or at least stopping the disease from getting worse. With that, the last thing I want to do is shout out a lot of the members of both the labs that I work in, as well as all of our collaborators who made a lot of the work possible, from various labs, as well as the Gladstone Institutes where I did a lot of this work. We have a lot of cores who enable us to do like the stem cell differentiations and all that fun stuff, as well as the funding that allows us to do all the fun work. So I will stop sharing now, and we can take all the questions that we were saving for the end. Dr. Dave?
Okay, can you hear me okay?
Yep.
Good. I saw that you were doing some CRISPR work there at the Lawrence Hall of Sciences molecule. Good work there. All right, we have quite a few excellent questions here. Pick up the first one. Person wanted to know how taking a vaccine like one for COVID affects DNA, mRNA, both, and is that related to editing genes?
DNA or mRNA what, sorry?
So how does a vaccine like the one for COVID affect our DNA, mRNA, or both, if it affects it, and is that related to editing genes?
From my understanding, it shouldn't. Even if it's an mRNA vaccine, like the idea of delivering, mRNA or protein is that these are things that degrade over time. So unless something really, really weird is happening, these things should go away over time. Yeah. So I wouldn't expect it to affect our genes in the same way that like a gene editing therapy would.
Okay, so the next one, this was an excellent question. It says, how do you get a strand? You talked about editing a strand. How would you get a strand on your own without it falling apart? Seems like it would be very fragile.
That's a good question. I don't do that work. There are I usually work in cells. There are people who do these experiments outside of cells, just like a strand of DNA in a dish. I think that DNA, unlike RNA, is actually pretty stable. And I know like we regularly order sequences of DNA that people make. They just like give us a strand of DNA that's really long. So I assume that technology is like has been engineered to a point where you can just like either make that DNA or order that DNA, and it should be pretty stable. I don't know the limits of like how long that can be reliably done or anything, but yes, that's not. I usually work only in cells. Good question.
Next question: would it be possible for CRISPR gene editing to be done in a human embryo?
Possible, yes. I would say, great question. Thank you for asking. Possible, yes. I would say that the majority of gene editing scientists, especially the ones that we work with, would say that at least for the foreseeable future, that's probably not something that we should be doing. My personal opinion is that I can't see a situation long-term where that would necessarily be the best mode of action. And there's a couple of technical reasons for this, but I guess I'll give you both sides of the argument. The reason people would propose this is that, as I said, there's a chance that in certain diseases, depending on how late you intervene, you could be more or less likely to be able to cure the disease by doing the gene editing. So the idea here is that people have is if you edit at the embryo stage when the entire person is just a few cells, you're very likely to edit all of their cells. So you'll probably have a really high efficiency of editing, and you're doing it at a time before the disease has had time to develop. The drawback, though, is that when you're editing at a stage in the embryo, when the entire person is a lot of cells, editing every cell means that you're also editing a lot of the sex cells, which means that the cells that will pass on those changes to future generations. So the risk here is that, if you accidentally have an off-target edit, like an unintended edit that appeared somewhere else other than what you were trying to do, or even if the edit that you were trying to make had unintended consequences, you're potentially subjecting future generations to that as well, not just yourself. So imagine taking a drug where the side effects affect not only you, but like potentially all of your descendants as well. So that's the huge drawback. Also, I think there are a lot of potential side effects that one could think of. And if possible, it would be better to limit that to only the kind of cells that you want to be editing, right? If it's a neuron disease and if it's a really like deadly neuron disease, there's no other treatment options, maybe you're okay with risking some potential side effects to keep you alive, right? But you maybe only want to be editing the neurons that you need, the cells that you need to do, that you need to edit to make that therapy curative, rather than editing every cell in the body and subjecting every cell in the body to those side effects. My technical answer is, I think if someone wanted to do embryo stage editing, they would need to do in vitro fertilization in order to do that. And at that stage, I think if you've done IVF and you've sequenced the embryo to know that it has the disease, my personal opinion is, like, in most cases, there's probably at least another embryo that, or maybe you could try again and like get another embryo that doesn't have the disease, rather than taking an embryo that does have the disease, subjecting it to all of these risks, and then turning that into a person, right? So I think personally there might be better options than embryo stage editing, but for the foreseeable future, I think most scientists would say that we should be focusing on not embryo stage, maybe in children, maybe in adults, but just not in the embryo stage.
Okay, so a couple more questions. This is a real good one. It said, could CRISPR be used to stop the progression of a prion disease in the future?
Yeah, I think so. I think that's an exciting application as well. Yeah, basically that definitely would fall in some of those filters that I talked about before. Like that's an example of a disease where it's caused by a problem protein. So you could imagine turning off that problem protein to stop the progression of the disease.
So you may have kind of answered this next one. It says, can the cells replace the missing dominant gene with a recessive gene from the other parent, and then sort of repair for the missing gene in that fashion?
I think maybe a way to reword that question would be, if you're missing the gene because you inactivated it, can you use the other gene that you already, if you're missing one copy of the gene because you've inactivated it and you're only left with one, can the one that's left make up for the one that you inactivated? I think there's a possibility that it could, that the cell could compensate in some way by, like, maybe since there's only one copy left, maybe read it twice as often to produce twice as much of that protein. It's possible. I think that probably varies on gene to gene and target to target.
Next one, it said, is CRISPR helping or advancing cancer research? Could it possibly help?
Yeah, there are a lot of people at UCSF, and I'm sure at lots of other institutes, using CRISPR to study cancer, to unveil new therapies. Like I said, even if CRISPR never makes it into a single patient as a therapy, it's already been super transformative for just studying biology. You can turn off a bunch of different genes and see which ones allow you to kill a cancer cell, right? And then design a drug to target that same gene. So there's lots of different ways that CRISPR and Cas9 have been used to study lots of different diseases.
How would you see the potential therapy being administered? Would it be a shot, a liquid to drink, a transfusion?
Great question. So this is the delivery question. How do you deliver this drug? And that I think also varies depending on what cells you're trying to edit. I think for some cell types, like I mentioned, the blood cells, I think there are ways to just like take them out of the person and then like zap them and get the stuff in, and then put them back. For neurons, it's a little bit more challenging. One of the ways that people have solved this problem, how do we get things into neurons, is they've engineered safer versions of viruses that are really good at getting into neurons, replaced all of the virus's own genetic information with instead information that encodes the Cas9 and the guide RNA, and using that to get in. We're hoping to develop more like non-viral ways of getting things in. So like maybe a nanoparticle that carries the Cas9 and gets it in. How you deliver that would probably be, for these brain diseases, maybe like an injection, either into the blood or into the cerebrospinal fluid, like in the spinal cord or something. Why am I pointing here? In the cerebrospinal fluid, or maybe into like the ventricles of the brain or something like that.
Looks like we have two more questions. One is, do you think that CRISPR will be able to be used for neurofibromatosis, since this disease has different types? So could we use it for neurofibromatosis?
I'm not familiar enough with that disease to comment on it, but I would say like we'd probably go through a similar series of filters, right, where we're checking all of these things of like just because there's different types of it doesn't necessarily mean it's not treatable, right? You could pick a type, a form of it that is monogenic and fits all of those things, but I don't know enough about that particular disease. Sorry.
And the last question is, where do you see the technology in 10 years?
Oh, well, hopefully people are being treated in a safe way, and some of these incurable diseases can be cured. Like I think a lot of the diseases that I mentioned, like ALS, for example, the median time of survival that someone has after diagnosis is so short that I think some of those diseases, or like Huntington's disease or sickle cell disease, I think are some of the first targets that people will be maybe going after, especially sickle cell disease and maybe Huntington's, and some diseases that affect the eye as well. So hopefully in the next 5 to 10 years, we'll see some of those therapies, and we'll have a lot of information on how safe it is, how effective it is, and whether we can start expanding into a lot of other diseases.
I did have a question that I missed up here. It said, is there an animal model developed for any of this kind of work?
Yeah, so for a lot of these diseases, people have animal models to study them. Yeah.
That's all I have on my list.
Cool.
All right, so I wanted to thank Gokul Ramadoss again for a really informative talk. It seems like everybody was really getting a lot out of it. You could tell by all the different questions, and even there were other comments other than the questions saying like, oh, this is interesting, oh, it's exciting. So yeah, we really appreciate you coming virtually and giving a talk here for us at MJC. And the recording will be available afterwards for viewing. There were several questions about that. So we can go back and review some of the topics that we've learned if we didn't take enough notes. And so I hope that everybody does that, and definitely spread the word and let your friends know about these other MAPS talks that are coming up throughout the semester. We hope to see you again and see more of you and more of your friends and other students at some of our future talks. And so thank you for all the committee for all your work, and thank you again, Gokul Ramadoss.
Thank you.